Welcome back. It's Bio 121, and we're looking at now the modern understanding of inheritance. And what you're going to see is all that work that we did to understand how meiosis occurs actually is going to turn out to be really important. And that's because the transition between, say, what happened when Mendel first published that paper back in 1866, right? The work wasn't touched again till 1900, as I said previously. Now, the people who did this work, their names aren't important. I suppose if you were those people, the names are important. But what I want you to see is, because it hadn't been touched in a period of time, 34 years or something like that, right? When people actually started looking at it again, what they tried to do is repeat what Mendel had done. Sign of a good scientist. If you can follow what he did, remember I made a big deal out of the fact that he kept really rigorous notes. You can see that what these guys are going to do is they're going to develop what it meant to be some sort of granular-like structure that will end up being defined as a gene. Okay? And even though that may not seem like a big deal, we're talking back in 1900. Nobody had yet figured out how to describe how materials are actually going to pass from one generation to the next. We also come to this concept that may be really easy for us in our fluent language to think about, but if we think about what mutation actually means, there's still no way to actually see what's happening at the individual nucleotide level at this point in time in life, right? So go back to 1900 inside of your mind's eye if you can, and you're going to see that the concept of a mutation becomes a very important idea. So Mendel's findings were based on statistical results, right? Remember, we created these Punnett squares and we guessed what was going to happen, and they were actually conceptual deductions. And those deductions, when you're looking at a simple system like snapdragons or peas or something like that, the idea, of course, that those hereditary particles could actually work was fantastic. It was spot on. But now what happened, by the time we actually begin to use new technology, the microscope, okay, you can actually see how the cells are changing as they develop through these different states. And what ended up happening, of course, is you end up seeing these structures here. These structures actually move. They appeared to behave in accordance with Mendel's observations. In as much, what this means is that these could be the hereditary particles that were responsible then for the different genotypes and then ultimately phenotypes, or phenotypes that we presumed had a genotype that supported them. You can actually take a look here and see that this is fluorescein, so tricolored modifications of chromosomes that have been stained. And you can see it's 25 micrometers in size here when you're looking at dollars to donuts. This is a Drosophilia salivary gland. And what you're looking at, of course, is the thread-like nature, and each of these banding patterns is going to be the repository for that genetic information. Now, even though we know this now, what I'm asking you to do is start thinking about what it might be like in 1900, when we're trying to figure out, well, what the heck is going on? And the reason why I pointed out this is part of the salivary gland from a Drosophilia is you're going to see that that was the model organism that actually helped us leap into our now current understanding of how this works. This gets us to this idea of what chromosomal theory of inheritance is, and these are the two individuals who are responsible for it, which states that chromosomes carry the hereditary information. So that means if you want to see a person who looks like they have male pattern balding, they have a specific eye structure, their bones have a specific structure, the informations that are carried on a subset of his genes would create now that phenotype. How this actually works is that during, meiosis, chromosome pairs migrate as discrete structures, and then each parent synthesizes gametes that contain only half of their chromosomal complement. And what I mean by this is, remember, this is a switch from a diploid structure to a haploid structure. Parent is diploid. The egg, the gamete that's coming from this, is the haploid structure. And what happens, of course, is during fertilization, the formation of that zygote, you now go back to the diploid number. So this is what I mean by this is partially review. And because you understand what meiosis is doing, you've recombined that genome, you can now begin to think about what Mother Nature might be doing with that information. If we follow this along, working with sea urchins and grasshoppers, what they did is they independently demonstrated that chromosomes occur in matched maternal and paternal pairs in meiosis. And what's going to happen is it's going to take T.H. Morgan, working in Massachusetts at the Marine Biological Labs at the time, I believe it's like 1904 now when this happens. What they do is they begin to look at how the genes are actually linked for the first real proof of a chromosome role in heredity. So here's a Drosophila, here's the Drosophila melanogaster, and here are meiotic cells, and you can see the karyotype before that, versus, say, this false colored image of a human karyotype where you have 22 pair of autosomes and 1 pair of sex chromosomes. In fact, it is Thomas Hunt Morgan who actually is responsible for identifying the fact that there is an X and a Y chromosome. At the time, this had not yet been discovered, and this is one of the major contributions to science that he actually adds. Those sex-linked chromosomes, you might recall earlier when we had graded expression of different eye colors, okay? That travels with the X chromosome, and here you can see the white eye, and that is a red eye. But remember, in previous slides, or previous lecture of materials inside your textbook, what I demonstrated here is, depending upon the gene dosage effects, okay, you could actually get some in-between chromatic distinction of how the eyes are pigmented. Why do we care? Well, we care because that linkage is broken up into one very important idea, and that is Morgan's work showed that sex linkage for a given trait were inherited together, which meant that they did not segregate independently, which is Mendel's second law. Okay, so when you go back to thinking about what Mendel did, is he said, look, all of these independent structures, color, seed coat, color, and roughness and smoothness, snapdragons and what they were doing, essentially he said that everything segregated independently, and this demonstrated that in fact that did not happen. More importantly, far apart traits could segregate independently, while as those genes or those pieces of information that were close to one another did not always separate independently. So there's going to be some explanation now for why we can actually see what was going to ultimately end up being discovered in synapses and homologous recombination in the future. Those crossing overs between homologous chromosomes become the mechanism that segregated genes on the same chromosome will actually work together. Okay? Now you can figure out the rate of this. Okay, and inside this really, really busy figure I'm about to show you, what you can see here is inheritance patterns of linked and unlinked genes. When you're looking at this inside the textbook, ask yourself this question. How would I set up this experiment so that I would actually see some of these outcomes? And it's going to take a little bit of time for you to figure out what they're doing. But what I want you to do is just simply say, oh, okay, if I start at the bottom here and I see that there's a parental and a recombinant and a recombinant, and I follow these backwards, what can I actually see in each of the independent figures? What is distinct about each of these constructions? You'll see where it's leading in a minute. Where it's leading in a minute, of course, is something that I showed you in lecture yesterday, but also in the pre-recorded class lectures, where the recombination events that actually create now the distinctly mixed populations of genes would now suggest that whatever's happened, however it's happened, you now have whatever each of these chromosomes are, they are carrying distinctly different genetic information. And this is occurring during, now we're specifically calling it, the synapsis of meiosis 1, which happens in prophase 1. And you can actually map this. Okay? One of Morgan's students, Alfred Sturtevant, he was an undergrad at the time, back in 1913, long since dead. But the idea, of course, is that if you're curious enough and you're motivated enough, it doesn't matter where you where you're going to school. In fact, I think Alfred reached out to Morgan and said something along the lines of, hey, I'm interested in looking at this. Do you have any space in my lab? And the answer, of course, was yes. And, you went and you did the work. And this is what you actually ended up doing, is he created a frequency of separations here. And what that means is the recombination frequency was a function of the distance between the genes. So recombination of 1% was chosen to be essentially equal to 1, what would ultimately be called centimorgan. And we don't teach you how to calculate these, which is in some ways unfortunate. But if we had a Drosophila lab, you could literally begin to figure out the distance between the mutations inside different organisms. Specifically, in this case, specifically inside the Drosophila, where it's easy to see. So here's that human karyotype again, and this is a karyotype of a human female, which means you have two XXs here, right? And the human male, of course, would have an XY. And I'm just simply trying to reiterate to you that what a geneticist does, okay, is they collect all this information. Notice there's a banding pattern here. You see this repeating pattern that we can use as a specific piece of information. Number one. Number two, they're artificially aligned like this. What they really look like when you look at them the first time is they look something like that. You have put false coloring on them, and then you rearrange them manually so that they actually end up looking like this, so you can make sense of what's going on. When they're aligned like this, you can say, oh, okay, they all look like they're all roughly the same length. Doesn't look like anyone is missing any one of these banding patterns. This is like, maybe there's something missing here. Look at that. Okay? This is definitely where you start playing this game. One of these does not look like the other, right? And what you do is you just simply go through it. And like, okay, there might be some explanation for what I'm actually seeing. And what you're looking for are these non-disjunction events. These non-disjunction events actually cause the rearrangement such that something is missing. Remember, I just said, I'm just looking at this quickly, right? What did I see? I saw that there was something missing inside 7 here. Looks like there's a darker staining pattern here on 8. So these things you would actually just look at a little bit more closely. Notice that green is essentially missing from the top there. Now, non-disjunction, take a look right here at 21. There's something missing there. I mean, completely missing. I mean, this is night and day. Okay, and that non-disjunction, the example we're about to work through, of course, is how, what could actually happen. Or if at meiosis or mitosis when cells are replicating, and you actually change the number of mutations or chromosomal arms that are sent between each cell, you can actually create problems. And this is trisomy 21. And this is what actually happens as a result from non-disjunction. And this is if a mother's age begins to go along for some period of time, okay? The frequency earlier in age of a disjunction resulting in a Down syndrome correlation, okay, is actually fairly low. However, as you age, as the mother ages, the meiotic consequence or the mitotic consequence of putting together that 21st chromosome actually seems to increase the correlation of the likelihood that Down syndrome would be the actual effect, meaning this is what you would see in the progeny. Now, for better or worse, nobody knows actually what's going on here in terms of a mechanism, but it's a repeating phenotype that you can actually see. And you can see that this was published in the American Family Physician back in 2000. So in the 20 years, almost to the month, right? This hasn't changed. In fact, if anything's been refined, is that the oocytes are now being stored in a frozen state. And I'm not actually quite sure what this does, what that does to this map. I think that would be actually a pretty interesting question to try and solve. You can watch this if you want. We've already walked through what actually happens for meiosis. You've seen it multiple times. Instead, what I want to think about is what are the effects of this and how could you actually see it? When we're looking here at sex chromosome non-disjunction, let's imagine, say, coat color for cats. In cats, the gene for coat color is located on the X chromosome. So in embryonic development, when one or two of the cat chromosomes is randomly inactivated, we'll come to how that happens, okay, in each cell, resulting in a tortoiseshell pattern, okay? If the cat has two different alleles for coat color, which is kind of cool. That's why you get the mottled sort of look to it. However, male cats only have one X chromosome, and therefore it never has a tortoiseshell color. So this is, once you understand that, this is one of those tools when you're looking at the outside world and going, okay, I see tortoiseshell, therefore I know that's a female, okay? Because you actually will never see this expression pattern on a Y chromosome. So how does this all work? It works because when something is being rearranged, it can be either pericentric or paracentric inversion mutations. These pericentric inversions include the centromere, and paracentric inversions do not. So take a look at, this is a normal chromosome, A, B, C, D, E, and F. And what you can see here, as we're coming to the end, is that you can switch around the actual centromere. Now, sometimes this doesn't cause a problem at all. Sometimes it does. And look at what actually happened here. C and D stayed in the correct locations, right? But look what happened to E and F. In many cases, this changes the shape, literally the size of the chromosome itself, where the pericentric can change. So here, pericentric can change the relative length of the chromosomal arms, where paracentric, on the whole, does not. It's simply an inversion or a switching of the cassettes of information. So, how does this homologous pairing actually occur? Well, when one chromosome undergoes an inversion but the other doesn't, this is what's going to cause an inverted loop. And this inverted loop is going to be something that's going to be like a hairpin. And that hairpin, okay, the inversion pairing is essential for maintaining the alignment and allows for the recombination. Now, at some point in time in the synapsis, obviously something went wrong here. Maybe it flipped in the other direction, okay, and that impingement caused that inversion that was occurring there. Now, ultimately, at the end of the day, we care about this because reciprocal translocations and non-homologous chromosomes can exchange material. And so what we're looking at here is a reciprocal chromosome translocation occurs. Take a look at what's happening before translocation here and after translocation from one chromosome to another non-homologous chromosome. And the reason why we're talking about this is because it's thought that this is one of the major mechanisms that's required for the expansion of the genome that allows us to have many of the genetic genotypes that we actually have right now, and also the phenotypes, such that we can increase the complexity because we have the multiple sets of instructions for every single cell to function appropriately. So that finishes chapter 13. Think of it more as a, hey, we're building on our understanding of meiosis so that when we start thinking about what transcription and translation are, or how DNA is packaged, this all makes much more sense. Thank you.